Click for IB Biology. Here we're looking at the compartmentalization of eukaryotic cells, in particular the nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, and ribosomes as we go. There is a blank PDF of this presentation down in the description so you can make your own notes, and then there's some 10 questions with answers so that you can check your understanding at the end. So here we're dealing with eukaryotic ultracell structures seen with the electron microscope with its high resolution. Take a look at that video on electron microscopy if you haven't already. And students will be familiar with diagrams like these, but these diagrams can never do credit to the complexity, the true complexity of the internal structure of a cell with its organelles and its various structures which are there. However, what we do need to be able to do is to recognize these features as shown in the diagram here, but from the real images produced from electron microscopes called electron micrographs. So the student is expected to be able to identify organelles from electron micrographs. And so we'll practice that as we go through these next 3 videos. We should be thinking in terms of shape, and that will always give us a clue to function. And the shape will be modified in such a way as to promote that function, which of course we often think of as adaptation. The cell contents are compartmentalized. There are small containers within the cytoplasm, and of course we call these organelles, and they are recognizable by their shape. Think of it like this. In a chemistry laboratory, we will use different types of container to perform different types of chemistry. And each container isolates, restricts and controls the chemistry within that container. You can't mix it all together and hope that those reactions occur independently of each other. Cells are exactly the same. They have containers of particular biochemical reactions. We recognize them by their shape. This reminds us of their function and their adaptation, but the shapes are the organelles. So each of the features that we are going to go through and look at now is an organelle with specialized adaptations to specialized chemistry, which is isolated within that structure. So let's take a look at our first micrograph. So the first micrograph here, this large structure up in the corner here, this is a portion of a nucleus, and the nucleus, depending upon the resolution of the image and also the size of it, we can find that there are pores within the nuclear membrane. And in fact, I should really draw this, the nuclear membrane is actually double. So we have a double membrane in the nucleus like this. The nucleus is about 5 to 10 microns in diameter, depending upon its function. Double membrane around the outside. Pores in the membrane to allow molecules to go into and out of the nucleus. We'll learn about those molecules as we go through the course. Dense regions within the nucleus known as the Nucleolus. So a bit of a myth buster here. This is not where the chromosomes are concentrated. This is actually a region more associated with RNA synthesis rather than DNA. The DNA is out here in this region of the nucleus. So looking beyond the nucleus with its genetic material, we can see in this image these strange lines, and if we look closely we'll see that these lines are actually tubular structures, and those tubular structures have small dots on the outside, and this entire structure is called rough endoplasmic reticulum. So the endoplasmic reticulum is referring to the tubes, and the fact that it's rough refers to these granules which are on the outside. And of course you may know that the granules are actually structures called ribosomes. Now, ribosomes are actually a form of RNA, so it's a large molecule. So these molecules are stuck to the outside of these tubes. The tube's diameter is about 20 micrometers. And what happens is that proteins are built on the ribosome. So the function of a ribosome is to build proteins, protein synthesis. And then the proteins are placed into these reticulum tubes and packaged before being sent, and actually in the case of this case exported from the cells. So if you have rough endoplasmic reticulum, the proteins which are being manufactured are going to be used outside the cell, not inside. Contrast that with smooth endoplasmic reticulum here and free ribosomes here. So this is a different situation. So these ribosomes are in eukaryotic cells, so they are much larger than the prokaryotic 70S. These are 80S ribosomes. Each of these dots is referring to a ribosome where we get protein synthesis. Now, if the ribosomes are free, we use the phrase free to mean that they are not attached to endoplasmic reticulum. If they're free from the endoplasmic reticulum, then the proteins which are produced are going to be used in the cytoplasm of the cell itself. And these small granules you can see are about 25 to 30 nanometers in length. So that's 25 thousandths of a millimeter. Free ribosomes making proteins for internal use within the cell. This is endoplasmic reticulum without the ribosomes. We call this smooth endoplasmic reticulum. Diameter is about 1 micron. The purpose and function of these is lipid synthesis. So lipid, of course, are oils and fat molecules. And also they're involved in the breakdown of toxic molecules within the cell, such as processes such are called detoxification. So one would imagine if one looks at the human liver, you would find large amounts of smooth endoplasmic reticulum, since liver function includes the detoxification of molecules within our bodies that we find toxic. So that's the nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, ribosomes with structure and function. In the next video, we'll go on to look at other organelles such as chloroplasts, mitochondria. The playlist covers the entire list of eukaryotic cell structures, and of course, there's a link to the channel.